This media was gratuitously produced by Clear Contributions Audio Video. The intellectual property contained within belongs to Dr. Richard Gardner and Garland Science. Clear Contributions. Let's have clarity.
All right, if we can just go over this this test real quick. Number one, it's the big two main classes of receptors. Surface receptors are inside the cell receptors, and those surface receptors are G protein, ion channel, enzyme linked. Receptor tyrosine kinases are an enzyme. Steroid hormones are the ones that can cross inside the cell because they're hydrophobic, and so they're intracellular. And the 7 transmembrane segments are the G protein receptors. You can look over number 2. Most people got most of those. Number 3, hardly anybody got this one. GAPs are the ones that work with the monomeric G proteins. So RAN, RAC, and Rho are the monomeric G proteins. So look most directly there. Most people got most of number 5. Steroid hormones are probably a first messenger, not a second. The receptors, like the G protein receptor, is not an enzyme, nor is it a second messenger. It's a different thing entirely. Different kinds of answers you could have had for the next one. But what I was looking for on A was that G protein swap the entire GDP, get rid of it, and put a fresh GTP on. D, plant cells don't have collagen. That's an animal cell thing. Plant cells have cellulose. You can look over the rest of these. Most of these are, once you look over what I wrote here, it's pretty self-explanatory. Number 8 on the sarcomeres. Not a lot of people. Well, not as many people as a lot of people didn't get that one. Or they put something on there that I couldn't recognize. So pretty much the myosin goes between, and then the actin is attached to the Z discs. Number 9 is the 9 pairs plus 2 in the middle. So I did actually count how many pairs you put. And I caught somebody with like 8 pairs instead of 9. So number 11, again, the actin and microtubules are fun. They have different kinds of movement, treadmilling, dynamic instability. You could have put those as 2 separate things. They have different ends, they have motors, and intermediate filaments don't do any of those things. Between actin and microtubules, actin has ATP cap, microtubule has a GTP cap. Actin's not hollow, but microtubules are hollow. There's 3 kinds of tubulin, 1 kind of actin. There's someone else that put something else that was also valid, and I can't remember what it was. This is just what I came up with. Number 14, what I was really looking for was that figure where it had the picture of actin and all the different proteins that can work with that. Bundling, capping, cross-linking, sequestering, there's more of them. Although if you put myosin, that technically is correct. This is a protein that works with actin or tropomyosin, so I gave you that as well. And then 15 was the, and most people got most of 15 as well. Anyone have any general questions on that? All right, we're missing a few people. Let's see, how many people are we now missing? 2, 3, 5, 6, 7, 8, 9, 10, 11, 13. We're missing Kashan and Ryan, who isn't going to be here, and Spencer. I want to do a little business here, a little class business. It is proposed that we, never mind. It's that time of the semester. Next week is the last week of class, which is amazing. And so we need to have talks. I need to talk to each group about these papers they're presenting. And so over Thanksgiving break, you're supposed to read it all, and nobody did, so now you've got to do it. Am I right or am I right? And so you do want to read it at least once, but twice is better. The first time I would just kind of skim through and see what it's about. Then I'd go back through again, and this time look at each figure, and each figure can spend quite a bit of time just looking at it, try to figure it out what it means. I have some notes that I took on these for myself just so I can remember what was going on, and I will email them out today, hopefully. I've got on here, again, who's doing which paper. So Damon, Spencer, and Michaela, the mouse paper, Travis, Blake, Amy, the flip-based paper, Danny, Simon, and Kashan, the mitochondria Golgi paper, Shirley and Alyssa, the kinase cascade paper, Ryan, Michael, and Tanner, the anthrax paper, and Ben and Vance, the MAD1 paper. I think that's right. So I'm going to email my notes, and they're not nicely. They're all kind of just stream of thought a little bit. There's typos in there. Look over those if that helps, but don't just read those only. Look over the paper. When we meet, I want to first ask, do you read the paper? So that's part of your extra credit points is to actually have read the paper. These are difficult papers, as all these kind of papers are. They're not like a novel where you just sit there and turn the pages and get lost in time. And oh my gosh, where did the time go? And how did I turn this many pages without even noticing? It's very much put thought into it and think about it. The materials and methods, generally, you don't have to spend a lot of time on that. They're just all the details about, what molarity their buffers were. You don't have to worry about that, but mainly the results and the discussion. And the introduction usually has a lot of background stuff, some of which is useful and some of which is just fluff. And but some of that's it's not going to make much sense unless you're in the field and knew all the other papers and stuff. But go through the background and then kind of look through my notes. That might be helpful. So I have another sign-up sheet. This is actually the same sign-up sheet, and I know we don't have everyone here. So the most convenient way to have this work is to meet with everybody at once. So if your group is all here, how many groups? Let's see, Damon, Spencer's not here, so that group's not all here. Travis, Blake, and Amy, they're all here. Danny, Simon, and Kashan is all here. Shirley and Lisa is all here. And Ben and Vance is all here, and then Brian's missing. So if your group is here, I put on here sort of a times. This hopefully can try to meet this week. I didn't put tomorrow afternoon because I figured that might be too early. Although if someone's ready by tomorrow afternoon, I'll be around tomorrow afternoon. Wednesday, so it takes about half an hour probably. So Wednesday from like 1:30 to 2:30 be starting time. So right after my class ends at 1:20, we can start then, and 2:30 would end at 3. I've got a meeting at 3:30, so I kind of got to get ready for that on Wednesday. Thursday I have no classes, so all day, anytime during the day on Thursday, and then Friday after 1:30. And then next Monday after 1:30, and that's probably about as late as we want to go because we'll try to get some presentations going. We probably have 6. We could do 3 next week Wednesday and 3 next week Friday. But if someone wants to go on Monday, we could have someone go on Monday and get it over with. So I want to, if you have your group with you, can you find your group? And then I'll just put this here, and if you can sign up for a time, again, just pick a time that's kind of within these times, Wednesday, Friday, Thursday, Friday, Monday, and what your names are and kind of what time. Plan on like 30 minutes, think about your schedules, and then just come up to my office at that time and try not to stand me up. If you don't know what that means, it means like when you have a date and they don't show up, whatever. And we'll find it. My office doesn't have enough space, but we'll find somewhere we can all sit and talk about this. So here's this. I'll just leave it here. Just take 5 minutes. I just want to kind of get. I have to go back and reread all these too, and so I kind of need to know beforehand who's coming when, so I can kind of remember to read the paper first and what's going on in there. Ooh, that's a cool picture. You're also going to want to meet with the group and kind of go over who's going to kind of present what and who's going to be responsible for what. Someone can, cut and paste pictures out of the original article. Basically, we're going to be making PowerPoints, and you're going to show us the data. Again, you don't have to show every single figure, but we'll talk about that, which figures you might want to use, and maybe different. Someone can talk about this figure, someone else can talk about the next figure, and so forth. Next week, basically Wednesday and Friday, but if someone wants to go on Monday, we could have somebody go on Monday. So we can do as many as 3 in a day, because 15 minutes each, that's 45 minutes, we got a 50-minute class, as long as we don't have a lot of time between. In the past, we've had times, again, where people bring a laptop and it doesn't hook up to this, or it doesn't work with a, hopefully this time, you can go on your iPad and go to the little screen Apple TV thing and then just pop it up and opens up on there and it works, and then you can just use those and show us the figures. If your group is not all here today, we'll try to repeat this on Wednesday, or everyone else committed here. And then the next thing we need to do is pass a list around about who's going to present when, so everybody kind of knows what we're doing. All right. How many people are still waiting for the list? Is anyone else waiting for those lists afterwards? You guys? No one fell off the roof. We did our Christmas lights. Yeah, we did our Christmas lights. We go on the roof every year. We got a big star on the roof and everything, and we've done this for like the last 10 years. We have this star. It's made out of PVC pipes, and we just spray painted them green to match the shingles. And we got 3 of them. We put holes in the ends and tie them together like a triangle. 3 more make the other triangle. Put one triangle on top of the other triangle, so it makes like a Star of David. And then lash it all together with good old Boy Scout square lashings, wrap it with lights. And it's a really steep roof like this, so I got to get someone to climb up, hold on to the door or window. This is Bethany, because I used to do this, but I'm too heavy and I slide down. And my mom says every year, "Oh, that's such great lights you had last year, but you really don't need to do that again. It's good. You've done well in the past, and you don't need those outside lights anymore." Because she's always worried we're going to fall off the roof, And so I'm like, I'll think about it. And then we do it, and then I just texted her and said, we sort of accidentally put those lights back up. Sorry, but so yeah, if you guys want to come by and see it sometime at night, come after 5 o'clock and the lights turn on. We got all these lights. We got our trees lit in the front yard too, so it looks nice. Let's see, someone else. Now Brian's not here. Are you in his group?
Yeah.
Can you commit him to something?
Yeah, we have the same exact schedule, so.
All right.
Has anyone actually fallen off the roof?
Not yet.
Not yet. That's good.
These guys want it. No. This is not going to be good at all. We're having issues here. Here, I was just saying how this works, and it's not working. I keep hitting D10, and it keeps telling me to put the error code in for D108. It's in the wrong room. I can't. This is crazy. All right, D108. Cancel. 10.
There you go.
I don't know what. All right. So we have Thursday, Wednesday, and this is now. This Monday, this was going to be next Monday, is that correct? Like a week from now. Just make sure we're all on the same page here. And make sure nothing else is conflicting here. I think that's good. All right, I will try to remind everybody, but please, does everybody write down their times? So otherwise, I'll get stood up. All right, we're going to go back and talk about cell cycle again. Let's just kind of start a quick go back through what we did last week because it's been so long. Let's see, what day was that? That was a Friday, and then we had tests on Monday, and then we had another week. So yeah. Anyway, cell cycle, G1, S, G2, M, memorize that. M is mitosis, and so that's got all the different phases inside of that. Prophase, metaphase, anaphase, telophase. You can go pro-metaphase if you want an extra thing. Interphase is everything except for mitosis. So G1, S, and G2 is all interphase. Stages of mitosis kind of just go through the little figures, kind of what happens each stage. Prophase, prometaphase. Again, metaphase is where they're all lined up. Anaphase is where they separate. Those are two big ones to remember. And then telophase, cytokinesis, plant versus animals. Animal cells pinch, plant cells have to build a new wall. Cytokinesis is not officially part of mitosis. Mitosis is separate the chromosomes, make two new nuclei. That's mitosis. Cytokinesis is usually happening at the same time, but it's a separately controlled event because you can have mitosis without cytokinesis. And then we talked about flow cytometry. Again, there's various uses of it, but in this case, we're staining the DNA so it glows when you hit it with a laser light. And if your cells have replicated their DNA, they've been through S phase, they've got twice as much DNA, so twice as bright, and the detector can detect how much DNA, one cell at a time. They go through really fast, but one cell at a time. So after you count, say 10,000 cells, you get a graph like this that shows you how many cells have how much DNA. The cells with the lesser amount of DNA are in G1. They haven't replicated DNA yet. Cells with the more are in G2, and the cells in between are somewhere in S phase. They've copied some of it, but not all of it, so they got some medium amount of DNA. So you can see graphs like this, and we're going to come back to these later. How how many people have started reading these other papers yet for homework on Wednesday? So on Wednesday morning, we'll talk about those first three. Is it Hartwell's, Nurse's, and who's the other one? Masui's, I think. So again, Hartwell did his CDC screen. CDC, you know what CDC stands for? Besides that, cell division cycle. So he called all his mutants a cell division cycle mutants, and he's looking for mutants where they all stop in the same place in the cell cycle. That means that there's something that is wrong. The cells are, well, in this case, they stop in G1, so whatever was required to get them to go from G1 to S, that's what got mutated. And so you can then figure out what protein that is and then what that protein does to help the cells go from G1 to S where it's mutated here and they don't go. And these are conditional mutants, in this case temperature sensitive, which means again that at the low temperature everything works fine. At the high temperature, that's when the mutant phenotype kicks in, and that allows you to actually grow the cells and work with them. And then when you do your experiment, you just shift up the temperature and then they'll stop at that point. And so you're also reading about these experiments where they injected cytoplasma into cells, and you take cytoplasma out of a mitotic cell and inject it into another cell, and suddenly that cell goes into mitosis itself. So there's something in the cytoplasm of a mitotic cell that makes other cells go into mitosis. And this was originally, again, discovered in egg cells, and the egg cells are actually finishing meiosis when this happens, and that finishing meiosis, egg cells are weird, like in mammals. You get your first meiosis cell division, then the eggs just sort of stop there. And then after the sperm comes in, meiosis actually finishes, and they finish dividing. And anyway, that's called maturation. So they didn't know what it was that's in the cytoplasm that you can inject in there, but it's called maturation promotion factor or promoting factor. Later on, they discovered it doesn't only work with egg cells in meiosis, but also any cells in mitosis. So then it became called mitosis promoting factor. So either maturation or mitosis, and conveniently, they both start with the letter M. So either way, it's something promoting factor. It promotes meiosis, promotes mitosis. But nobody knew what it was, so they just called it MPF. And so for years and years, they talked about MPF, and everybody talked about it, but no one knew what it was. And then, of course, the other papers, we'll talk about our Weinert's checkpoint papers, where the cell monitors how things have gone in the past, and did we make a mistake in replicating DNA, or do we have damaged DNA? Are all the chromosomes not attached to the spindle? And if so, if any one of those problems happen, the cell stops, doesn't go through to the next step until it fixes it. So that's the idea of the checkpoint. So those are those papers we'll talk about. Okay, so then this gets us to talk about the main driver of the cell cycle. So they've analogized the cell cycle to an engine. What actually pushes a cell from one phase to the next phase to the next phase, and makes it go through from G1 to S and G2 to M? And the answer is the cyclin-dependent kinase. So it's kinase. So what does kinases do? They phosphorylate stuff. So it's a special kinase that phosphorylates stuff, and it's dependent on a molecule called cyclin. And you can kind of think of cyclin as like its allosteric activator. I don't think anyone ever calls it this, but that's pretty much what it is. It doesn't work unless cyclin is attached to it. And so they've discovered, and then you'll see in the cyclin paper we'll read later on by Hunt, he discovers cyclin, and cyclin during different cell cycles, as you watch the cells go from mitosis to interphase and then back to another mitosis, the cyclin goes up and then it goes back down, it goes up and then it goes back down. And that's why they call it cyclin, because it kind of goes up and down. So the levels of it's a protein, so you get more and more and more of it. And then right before mitosis, you see a lot of cyclin, and then suddenly all the cyclin disappears, and then the cells go into the interphase, so they're now leaving mitosis, going into G1. Remember, interphase is everything except mitosis. So if that's mitosis, G1, S, G2, so all of this is interphase. So in other words, where cells are leaving mitosis, going to interphase, they're right here going into G1, part of interphase. And the cyclins go down, and then they start building back up again. And so it was hypothesized that somehow these cyclins are what's driving the cell through different stages of the cell cycle, the levels of the cyclins. And then we also look at the mitotic CDK activity. CDK, again, is a cyclin-dependent kinase. So that's the big kinase. And its activity, meaning how, if it's on or off, That's the green thing here, and notice how it's off, 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 and suddenly it turns on right here. There's one. It just suddenly turns on, going from no activity to a whole bunch of activity. Stays on for a while, then suddenly it turns off. And then the next cell cycle, it suddenly turns on and goes off. And so basically what's going on is that the cyclin-dependent kinase needs the cyclin to work. You gotta have a certain threshold level of cyclins to get that on. And as the cyclin levels increase and increase and increase, now you've got this sort of threshold level that kind of can help the cyclin-dependent kinase turn on. And it turns on suddenly, drives the cell into mitosis, and then just as suddenly as it turned on, it turns back off again, and the cells are now let go of mitosis and they're back into G1. And so it's kind of controlled by these cyclin levels. And there's more controls we'll talk about in a minute. And what's important to understand is that the cyclin is the actual amount. On the graph here, it's the actual amount of cyclin. So the amount increases and then it suddenly decreases, and then it increases and it decreases. The CDK, the cyclin-dependent kinase, is not the amount of the enzyme. It's if it's on or off. The enzyme's always there, but it's either on or it's off. And so the green thing is on or off, whereas the red line is how much you've got, and then it goes away. So we'll come back and talk about how this is all controlled. So different stages of the cell cycle have different cyclins that can associate with the cyclin-dependent kinase. So for example, the cyclin-dependent kinase can drive the cell into S phase. And then you've got S cyclins that associate with it and give it that activity. It gives it different targets to phosphorylate. But then when it's time to go into mitosis, a different cyclin attaches to it and then drives it into mitosis. So it gives it different targets depending on which cyclin. So the S cyclins, again, go up and down. The M cyclins go up and down at different times of the cell cycle. And mostly in the chapter we'll be talking about mitosis, the control of mitosis, going in and out of mitosis, but a little bit on the S phase as well. So this is just a vertebrate chart here. Don't memorize all this, but it shows there's different cyclin-dependent kinases. There's kind of one that drive the cell into S phase, one to drive the cell into M phase, one to go from G1 to S, sort of, and different cyclin partners. And so in yeast, where a lot of this was first done, so higher eukaryotes, when I say higher eukaryotes, that means animals and plants, basically, and so that's animals and plants, versus, say, fungi, which is a lower eukaryote. Higher eukaryotes have multiple cyclin-dependent kinases and multiple cyclins. For each of the different stages of the cell cycle, but fungi has only one cyclin-dependent kinase. It does all the work for S phase and for M phase and everything else, and it still has a few cyclins, a few different cyclins, but not as many as vertebrates have. So it's kind of the same idea, but when you do this in yeast, you don't have to worry about all these different CDKs. There's just one CDK, and when it's attached to a S phase cyclin, it drives the cell into S phase, but when it's attached to an M phase cyclin, it drives the cell into mitosis. But then when you get into the higher, like the vertebrates and animals and so forth, then you've got multiple different kinases as well as multiple cyclins and even more cyclins, and this is one reason that yeast is a great organism. It's just simpler. So you can do more easy research on it without getting all bogged down into all the details of. All right, so we'll talk just a little bit about S phase, going into replicating the DNA, and this is pretty much the only slide that talks about that. And then after that, it's going to be all sort of mitotic stuff. The goal of S phase is to replicate your DNA and to do it once. You don't want to replicate it twice, you just want to replicate it once. So you have, every chromosome gets copied. If you replicate 2 or 3 times and you've got, that's weird, then, well, you could become polyploid that way. And that happens by accident sometimes, but you want to do it once and only once. So you want to be able to get the whole replication thing started, and then you want to prevent it from happening again until the next cell cycle. So here's your DNA. Where you're going to start replicating is the origin of replication. I think we've talked about origins of replication, have we not? We talked about replicating a little bubble that comes out. So your origin of replication, there's a whole bunch of proteins that sit on that called the origin recognition complex. And they sit on that origin, which is part of the DNA, and they get ready to start replicating. And one of those is CDC6. And again, CDC comes from Hartwell's screen. Although I don't remember if this is Hartwell's CDC6 or Nurse's CDC6. I think it's Hartwell's, but it doesn't matter. Anyway, there's this protein that's now called CDC6. And it's one he discovered, in either Hartwell or Nurse discovered in their big screen for the CDC mutants. And it sits on there, and it's part of that origin recognition complex. And that complex helps recruits the DNA helicase and gets it all set up to start replicating. It's called loaded. It's like you've got your gun cocked and it's all ready to fire, but it's not quite ready to go yet. But it's all, all the things are in place to start replicating your DNA. And then the S phase CDK comes along and phosphorylates some of those proteins on the origin recognition complex, and that kind of fires everything, gets it going, and starts the helicase going. So that kinase activity is what turns the whole thing on, and it begins to replicate. And what it doesn't show here is that and it should show, it used to show this, but the CDK not only phosphorylates some of those origin proteins and fires and gets the whole thing replicating, but it also phosphorylates CDC6. And when CDC6 is phosphorylated, it becomes vulnerable to proteases. And again, what are proteases? They degrade proteins. So your CDC6 becomes phosphorylated, and that makes it vulnerable to proteases, and they degrade it, and then you have no more CDC6. And that's why you can't replicate again. You can't set up an origin recognition complex without CDC6. So CDC6 gets destroyed, and it will not be created again until the next cell cycle. So then you're prevented from replicating twice. So the CDK comes along, gets the process started, and also destroys the CDC6 at the same time. So you can't do it again. Any questions on that? So it's got like a dual role here, to start things and to prevent them from doing again by the CDC6 issue. All right. So study that figure, and then if people have questions, we'll come back and do it again. So now, this is now more of the, well, this applies here to pretty much any time, either S phase or M phase. And it's about the cyclins themselves. So we talked about the cyclins themselves, which actually, like from here, they go up and they go down, they go up and they go down. They increase because you're just basically translating protein. You've got RNA and you're translating and making more and more protein. You keep translating more and more protein. Cyclin is a protein. And then it's suddenly destroyed, and that's where it goes down. So the cyclin gets destroyed, and it's getting destroyed by proteases as well. And so this gets destroyed when it's targeted for destruction by ubiquitin. So we just had the CDC6 that gets phosphorylated, that targets it for destruction. It's sort of like drawing a bullseye. It tells the proteases, destroy this target. Ubiquitin is a little protein molecule that when it's attached to something, it also targets it for destruction. It basically is a marker for the protease to recognize that and say, well, this is what you destroy. So the cell adds ubiquitin to the cyclins, and that targets it for destruction. The proteasome is like a little machine made out of proteases. So it's, instead of the proteases all just sort of loose and running around the cell, they're kind of all put into this little machine. And then that's sort of called the proteasome. It's like this destructive machine. But it's basically proteases. So the cyclin gets destroyed, and then the next cell cycle, it starts to build up again, and then it gets destroyed, and then it goes down again. And that's what kind of helps to drive the cell from one phase to another phase. All right. You will see the word ubiquitin, and when it's put on, there's the old word, well, depending on who you ask. Here they have the word ubiquitination, meaning to add ubiquitin to something. And you'll also see the word ubiquitination instead of ubiquitination. They mean the same thing. Why there's two words for the same thing, I don't know, but one's ubiquitination, one's ubiquitination. And I don't know if it's the authors of the book are Brits, and the British people like ubiquitination. I learned it as ubiquitination, so this one bothers me, but anyway, either way works. All right, so more complexity here. In order to have your CDK active, it's necessary to be complex to the cyclin, but that's not sufficient. You all know the thing about being necessary and not sufficient. So you've got to have, there's more to it than just that. You must have the cyclin on there. There's also an inhibitory phosphate that gets stuck onto the CDK, which inhibits it, and that has to be removed first. And so the the way that phosphate gets put on is by this kinase called the Wee1 kinase. So that puts on the phosphate, which inhibits the CDK. And then this phosphatase, the CDC25 phosphatase, and CDC25 I think is from Nurse's CDC scheme. That's what takes off the phosphate. And then the cycline's attached. And the point of this is you want, when it's time to go into mitosis, you want the cells to go into mitosis suddenly. You don't want them, so as the cycline's kind of building up gradually, you kind of don't want all your cells to kind of gradually think they're kind of in mitosis and not really. You want to just be yes or no. So that's why we want a really sudden rise in CDK activity. It's either on or it's off. And the cycline kind of gradually would tend to kind of make it kind of half on and half off, and that's not really good. So by having this other mechanism of the phosphate in there, you can basically have this inhibitory phosphate, and as your cycline rises and rises and rises, your CDK is still not on because that inhibitory phosphate is on. And then you can suddenly just remove that inhibitory phosphate, and that's what suddenly drives them into mitosis. So you've got the cycline all ready to go, there's enough of it there, and then you suddenly take off that phosphate, and bam, you're in mitosis suddenly. And then just as suddenly you degrade all the cycline, so that's where the cycline goes down, and that turns it off again and drives you back out of mitosis. So that's why there's this second method of controlling your CDK by using this phosphate. And so there's actually kind of a feedback loop. So here's our inactive cyclin-dependent kinase. It's now attached, start to build up the cyclin. Beginning there was no cyclin. It starts to build up until they've all got cyclin on them, but it's still inactive. And then this phosphatase takes off, the Wee1 kinase that's put these on, the phosphatase suddenly takes them off. And once it takes them off, this is now active. And once that's active, one of the things it does is to phosphorylate the phosphatase. That turns it on. So we have a little bit of phosphatase that's already on. That takes off the inhibitory phosphates. That activates the CDK. The active CDK then goes to the rest of the inactive phosphatases, inactive CDC25s, puts the phosphate on, which activates those. So it's like a positive feedback loop. And this happens really quick. So once you got some of these that are active, all you need is like one molecule of that CDC25 that's on, and the rest of them can all be off. And that one on will take off these phosphatases, activate this. This then goes and phosphorylates all the other CDC25s. It's an enzyme, so it can happen really fast. You can do thousands of reactions a second. Puts a phosphate group on those CDC25s, activates them all, that turns them on, and now they're all on, so they can go back and take off the phosphates of the other ones that aren't yet on, and that just makes it all happen suddenly. Does that make sense, everybody? So this whole feedback thing all happens really quick. Suddenly all the CDKs were off, and now suddenly they're all on really quick with this whole positive feedback thing. So this is a bit of detail. So you're gonna have to memorize that the CDC25 is the phosphatase, which pulls off the inhibitory phosphates. That the kinase actually activates the phosphatase by adding a phosphate to that. And that the Wee1 is the one that originally had put those phosphates on in the first place. And when you read Nurse's paper, how many people have read Nurse's paper already? So if you read Nurse's paper, they talk about the Wee1 kinase and why it's called Wee1. Wee means small in Scottish. He's a Brit guy. He's Scottish. Wee, like the wee babies, they're small. And the mutation in the Wee1 basically means that if you didn't have a Wee1, now you're not going to put that inhibitory phosphate on. That means the CDK will be active prematurely. And by being active prematurely, the cells will go into mitosis prematurely, and they'll divide when they're too small. They didn't get a chance to get big yet. They just suddenly go into mitosis prematurely, so they're small, and they're wee, because wee means small in Scottish. And that's why they called it Wee1. So when you read that, we'll go over this again on Wednesday. All right. So that's the main ways of controlling the kinase. The cyclin levels plus these inhibitory phosphates. There's one other way we're going to talk about that can control the kinase, and that is this molecule called P27, but it is a cyclin-dependent kinase inhibitor, or a CKI. So again, CDK is the cyclin-dependent kinase, and then you can have this, add the word inhibitor on it, and now you've got, that forms a CKI, which is the cyclin-dependent kinase inhibitor. So P27 is a cyclin-dependent kinase inhibitor. It just binds the whole thing and just basically blocks it from doing anything. It's pretty boring that way, no phosphorylation, where it just sticks to it and just kind of. So if you have that, you'll just kind of basically shut off all of its activity no matter what else happened, with or without the kinase, with or without the cyclin, with or without the phosphorylations. You stick this on here and it just turns it off. All right. So that's going to come back up again in a minute as well, or sometime as well. We'll come back to this in a minute. All right. All right, so getting ready to go into, or as you start to go into mitosis, you have to condense your chromosomes. The chromosomes were big thready things, and you want to condense them to where they look like this. Why is it important to condense your chromosomes before you go into mitosis? Say that again?
You don't lose genes.
Okay. Yeah, imagine that you had, remember these chromosomes can be several feet long inside of a cell, and they're all tangled up threads, and you're going to try to separate them, and they're just going to get tangled up. So you've got to like get them all compacted like this so you can easily separate them, and then you can let them unwind again. Now you don't want to keep them like this, then you can't ever replicate or transcribe from that. It's all tightly wadded together. But I will just put in Christmas tree lights up, and I'm trying to wrap around the branches, and the easiest way to wrap them is to get them all in a big wad, and then I can take the whole wad and just throw it over the branch, take it out and throw it over the branch. Now, if it's all tangled, then I've got to kind of do all this thing. So it's the same idea. We're gonna condense everything, and then you can easily separate, then you can uncondense. These become 10,000 times shorter when you condense them. So imagine 2 miles is about 10,000 feet, and I hate to use miles and feet because we're supposed to be metric in science, but imagine 2 miles is 10,000 feet, and to condense a rope that's 2 miles long, now it's going to become 1 foot long. That's how much these things are condensed. That's just amazing. So they're super thin, obviously. All right, so we've got what's called cohesins and condensins, and they both are very similar molecules. They're little protein rings, and they're kind of like handcuffs. The condensins basically hold little loops of DNA in the little handcuffs like this, so they kind of help to condense the DNA by kind of helping it to loop and then kind of holding the loops together. And then the cohesins are basically holding the sister chromatids together. So once you copy your DNA, you've got each chromosome is duplicated, you've got 2 long pieces of DNA, there's the sister chromatids, you've got to hold them together, and that's the cohesin rings that are holding those together. You don't want to just let them drift loose, because then how are you going to make sure each cell gets one? You've got to have them together until it's time to pull them apart. So that's the cohesins and the condensins. So they're probably evolutionarily related proteins because they kind of have the same shape and the same, or very similar types of functions. Cohesins and condensins, here's your 10,000 times shorter. All right, so here's anaphase happening, and your spindle set up and attached to the chromosomes, and then pull the sister chromatids apart. And then right here you've got, this is more of an animal type of cytokinesis. Actin is basically wound around the cell. Actin is what is going to kind of pinch the cells, and then the microtubules are doing their job with the spindle. All right, all right, so this is basically the spindle sort of formation going on here. Again, the centrosome is an animal sort of version of the microtubule organizing center. So it's sort of similar in other organisms as well, but you kind of have to form this spindle and then attach to the chromosomes. Let's not spend too much time on that one. A little more on this one. As we separate the chromosomes, we've got some, I thought I had another picture of this, but we have some, here we go. Here's our spindle. Some of the microtubules are attached to the chromosomes. You can see some attached to the chromosomes. Some are not attached to the chromosomes, but they are kind of overlapping with each other right here. I get a little dot here. Come on, man, there's one little dot. Here we go. These are overlapping with each other. And then some of them are going out the back end and attaching to the cell membrane on the inside. And then as anaphase progresses, 2 things have to happen. The sister chromatids have to separate from each other and go opposite directions. And then the entire spindle gets longer at the same time. And as the sister chromatids separate, it's sort of a pulling on them, but it's also a motor function. And I think this one, they might have taken out of the book. This one's from the previous version. But here's the kinetochore. So that's where the microtubules are attached to the chromosomes, and then the chromosome is actually the spindle fiber. The microtubule is actually degrading. At the same time, there's motor proteins, and this is like these yellow ones are like motor proteins, and they're pulling this whole thing that way. So it's sort of like those cool movies where you're like on a bridge and you're going really fast, and the bridge is kind of just collapsing behind you, and you have to go fast enough to keep up so that it doesn't collapse underneath. So the microtubule is collapsing while those motor proteins are moving down and pulling the chromosome down. So that's part of anaphase. Somewhere I had a better picture of, here we go, anaphase A and anaphase B. So we'll just end on this note. Anaphase, again, at the same time, simultaneous, and A and B are happening simultaneously. A is that the chromatids move apart, and B is that the entire spindle gets longer. So the motor proteins here are pulling the chromosomes to each side, and that's anaphase A. And then these motor proteins on these overlapping microtubules, which are not attached to chromosomes, are pushing each other apart and making the entire spindle longer, pushing the poles apart, and that's anaphase B. And they happen at the same time. The A and B sound like, oh, A happens and then B happens, but it's actually at the same time. So at the same time, the chromosomes are moving apart, certain motor proteins, other motor proteins are pushing the fibers apart, and the whole thing's getting longer. So that's the A and the B. All right, so Wednesday, we'll talk about those 3 papers, and we'll do more cell cycle stuff. And then Friday, we've got the other 2 papers, and then we'll do more cell cycle stuff.